Hello and welcome to this week's episode of Brandon Explains Basic Biopsychology. Today we're going to talk about the neuron.
In this image from Britannica Academic, you can see neurons in red supported by glial cells in green.
Whether or not a neuron fires is the result of the summation of postsynaptic potentials. These postsynaptic potentials can be either excitatory or inhibitory and are received on the dendrites or cell membrane surface of the neuron. If the summation of the excitatory and inhibitory postsynaptic potentials depolarizes the neuron past the threshold of excitation, which is about negative 65 millivolts, this will trigger an action potential response in the neuron. Now, unlike postsynaptic potentials, the action potential of a neuron is non-graded and non-decremental, meaning that it is all or none, and there is no degradation of the signal while it travels along the axon.
Here is another picture from Britannica Academic showing a nerve cell. What's labeled as nerve fibers are the dendrites of the cell, which receive the EPSP and IPSP potentials. These potentials then summate at the axon initial segment.
One kind of simple, silly analogy I like to use for action potential in a neuron is kind of how people deal with gossip. You know, if one person comes to you and says, somebody said something, about you, you might, just not really think about it, especially if somebody else said they had said something positive. It kind of cancels each other out. But if several people come to you and say somebody's saying something negative, or if several people come to you and say somebody's saying something positive, it influences your overall attitude to the point where if enough people say something, you might feel the need to go and speak to somebody and say, hey, I heard you're saying negative things, or hey, thank you for saying all the wonderful things you've been saying about me. But the summation of the postsynaptic potentials, or rather the summation of the bits of information that an individual receives, influences whether or not they cross a threshold of action, whether or not I need to do something about all these bits of information I'm receiving. So when the summation of postsynaptic potentials crosses the threshold of excitation and significantly depolarizes the neuron, action potential is then generated from the axon initial segment. Once the action potential is generated in the axon initial segment, a sequence of voltage-activated ion pumps along the axon create a chain effect, a domino effect, where they activate each other in sequence very rapidly down the line of the axon. In a myelinated axon, this chain reaction happens more efficiently. The myelinated segments of the axon act as a slipstream, allowing the voltage to jump a segment of the axon and activate a voltage ion pump further down the line in the axon more efficiently.
Here you can see a comparison image between a myelinated and an unmyelinated axon. The action potential of the unmyelinated axon has to hit every voltage-activated ion pump along the axon, where in the myelinated axon, the voltage-activated pumps at the nodes of Ranvier are the only ones that need to be activated, hence the increase in efficiency.
Myelination occurs and is increased with repeated usage of certain neural circuits. So myelination is a part of neuroplasticity as efficiencies among certain neural circuits are improved. And we'll get to glial cells and their role in this in a little bit later, but this is part of how glial cells serve and support the neural network. So whether the neuron had a myelinated or an unmyelinated axon, or even if it had no axon at all, when an action potential is generated, it's going to move to synapses. And these synapses are going to either be electrical or chemical synapses. Electrical synapses are also referred to as gap synapses or gap junctions. If the action potential reaches a chemical synapse, the voltage will activate ion pumps that will bring in calcium ions to the button on the end of the axon and will cause the vesicles to fuse with the membrane of the button, releasing neurotransmitters into the synaptic gap between one neuron and the transmitting neuron and the receiving neuron. Once a neurotransmitter has been released into a synaptic junction, the process begins again as the neurotransmitter will either generate an EPSP or an IPSP in the neuron or neurons to which it is transmitting. In an electrical synapse or a gap junction, neurons are directly contacting one another. The action potential is transmitted directly as a voltage. So the ion pumps of one neuron are directly stimulated along that domino effect chain of events from the voltage of the previous neuron. So this is a much more direct, faster form of synaptic transmission between neurons, and it's typically used to coordinate activities between groups of cells for more limited periods of time. There are two analogies that I find useful when comparing electrical and chemical synaptic function. For chemical synaptic function, I like to think of kind of a telephone tree, which was a concept a little more prevalent before cell phones. In a telephone tree, one person would be responsible for passing news along to two or more other individuals. Those individuals would then be responsible for passing information on to two or more additional individuals. So within 3 or 4 tiers of a telephone tree, all the members of an organization could be notified relatively quickly about changes or news that was important. Now, for electrical synaptic transmission, I think that urgent news weather broadcasts are a more useful analogy. Urgent news broadcasts, when there's a storm or a tornado warning, they're pushed directly to every news outlet, every radio station, every cell phone, every form of communication in the area to urgently organize people to take important and immediate action. It's a system that has to be used more sparingly, but it is definitely faster, and it has a greater sense of urgency and overall organization. Moving on from synaptic transmission, neurons are not the only cells that make up the central and peripheral nervous system. Neurons are balanced and supported in about a 50-50 ratio with other cells called glial cells. Now, glial meant glue, roughly, because originally glial cells were thought to just kind of be structural cells that supported the neurons. But that has changed recently. In fact, there are 9 types of glial cells that have been identified, and most of them are not well understood. However, there are 4 types of glial cells that have been fairly well researched in the recent decades. Those 4 types of glia are oligodendrocytes, Schwann cells, microglia, and astrocytes. Let's take a deeper look at each of those. Oligodendrocytes are present only in the central nervous system. They help myelinate axons, and not just of 1 or 2 segments of a single neuron. They can myelinate the axons of several neurons simultaneously, which seems to increase how the neurons wire together and especially how efficiently the action potentials of one neuron transmit to the next. Schwann cells serve a similar function to oligodendrocytes, but for the peripheral nervous system. Now, differently than oligodendrocytes, Schwann cells can only myelinate a single segment of an axon at a time. So, while they are not capable of coordinating the synaptic connections between neurons to the same extent that oligodendrocytes can, Schwann cells have a unique advantage. Schwann cells can direct repair of neuronal circuitry. So this is why repair of neuronal damage in the central nervous system is not possible in mammalians, where the peripheral nervous system can be repaired comparatively well. Schwann cells serve a similar function to oligodendrocytes, but for the peripheral nervous system. Now, differently than oligodendrocytes, Schwann cells can only myelinate a single segment of an axon at a time. So, while they are not capable of coordinating the synaptic connections between neurons to the same extent that oligodendrocytes can, Schwann cells have a unique advantage. Schwann cells can direct repair of neuronal circuitry. So this is why repair of neuronal damage in the central nervous system is not possible in mammalians, where the peripheral nervous system can be repaired comparatively well. The third type of glial cell, microglia, are the housekeepers and demolition regulators of the nervous system. They function in both the central nervous system and peripheral nervous system, and their function is to get rid of waste and help regulate the lifespan of neurons. The third type of glial cell, microglia, are the housekeepers and demolition regulators of the nervous system. They function in both the central nervous system and peripheral nervous system, and their function is to get rid of waste and help regulate the lifespan of neurons. Lastly, astrocytes are the largest and, in my view, the most interesting of the glial cells. Astrocytes intersect with neuronal cells as well as the neural blood vessels. So astrocytes actually direct and influence vascular dilation and constriction in the brain where blood flows, which is, of course, one of the key indicators we use during brain scans to identify what region of the brain is in function or being used during a particular process. Astrocytes also influence neuronal connection, and astrocytes connect to one another. Like cells bonding to like cells. There are gap junctions between astrocytes that will help astrocytes conduct tasks in unison. Interestingly enough, also, astrocytes are evenly distributed in regions where they occur in the brain. So they don't overlap and they don't bunch up together. They're very orderly and very structured, simultaneously acting as scaffolding, but also conducting, especially the flow of neuronal synapses and blood. Astrocytes also serve part of a function as, or sort of supplement the blood-brain barrier, allowing certain molecules to pass from blood vessels into neurons and preventing certain molecules from passing from blood vessels into neurons. So astrocytes are definitely the more interesting, in my opinion, of the glial cells that we currently understand. Researchers have determined that the more intelligent a species is, the more glial cells are present. And the correlation between glial cells and intelligence has been supported by some of the research with astrocytes. So my personal hypothesis is that glial cells serve a role in consciousness, with the microtubule gap junctions that organize evenly distributed astrocytes, and these astrocytes are responsible for synaptic pruning and for the direction of molecules and nutrients and the constriction and dilation of blood vessels. It makes sense that with all these coordinating responsibilities and this high use of energy and the yin and yang balance, there's a parsimonious balance between neurons and glial cells in this sort of 50-50 configuration in the brain, this interplay that I think probably leads to the understanding of the self in a higher intelligent species. And with that, we'll conclude this week's edition of Brandon Explains Basic Biopsychology. If you liked the video, please subscribe and comment below.